All right. Hello and welcome to today's episode of the Method Made podcast, where we discuss technology transfer challenges for cell and gene therapy. Today, I'm joined by Arnaud Deladeriere. Welcome, Arnaud. How are you?
Yeah, I'm good. Thanks. Thanks for having me on it.
Yeah. So, Arnaud, you are a consultant. You have a lot of experience in cell and gene therapy in CMC. And in today's episode, I'd love to discuss with you the make or break manufacturing hurdles and why CMC and tech transfer might be an underestimated risk for new biotech, specifically in cell and gene therapy.
Yeah, absolutely. That's basically what I do for a living. So more than happy to talk about that and hopefully help whoever's listening.
Awesome. So maybe to start with, what exactly, like for founders, people who are at the earlier stages of their biotech journey, who might have just finished say like a PhD, what exactly is CMC?
Yeah. So think of the fact that whatever scientific discovery these academic labs, researchers, very young companies are doing, great. The moment I have something that I can potentially start a company with. Whatever discovery you make, if it's a therapeutic, will have to go in a patient at some point. Um, the idea of CMC is to basically transform that discovery into a product that will hopefully one day treat patients. But of course, you don't put a therapeutic product into a patient without a set of rules. And those are basically encompassed in the whole idea of CMC is you go from a small scale product that you may have done into a small flask or in a 6-well plate. Well, that's not enough for a patient, especially when you talk about like, let's say a CAR T. We can keep the idea of a CAR T as a thread and we can spin out for some specific products if we need to. But for the purpose of the podcast, maybe we can keep the CAR T as a thread because that's one of the major cell therapies right now.
Mm-hmm.
Um, you need a bag with a few billion cells to be infused in a patient. So you need to manufacture it at a certain scale that is much larger than what you've done so far. You need safety checks, contamination, um, whether the product actually has like is identified the way you think it needs to be identified. So like it's a CD4, CD8 product. It has the CAR that you put in it at a certain level. It doesn't have any bacteria contamination, doesn't have any mycoplasma contamination, all those factors. So you need to think through this. It's the set of rules that will basically define the product that you're going to into your patient. And that goes obviously with regulatory submissions. So you build that package to basically justify and show the regulatory agencies that you don't really just have a scientific discovery anymore. You have a patient product. And of course, like the M in CMC is manufacturing. You need to manufacture that product, whether you do it yourself or you do it through a CDMO. That product will have to be manufactured in a GMP environment to make sure that all those rules are followed and you have a safe product. Efficacious is a thing of the future for your phase two, your phase three. What you're really required to show in the first place is safety.
Okay. So CMC is like you need this to get to phase one is the general scope.
Yeah, you CMC is basically the bucket of work that you have to do to get to your clinical trials
Okay.
at
And
large.
yeah, okay. So, I understand the controls you've discussed and you need to manufacture it has to go into a bag. The last thing that'd be kind of nice, just what about the first C, the chemistry? What is that about?
So for us, it's kind of like the product, right? It's
Yeah.
what you need to generate, what you need to justify about the product that you're going to put into your patient.
Okay. Now, something that there's been a lot of discussion in the space about, and I think I've seen you mention this as well, is the product is the process. This concept and different
No,
places
no,
where that.
no, 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 no.
So what are your thoughts on this? Why not? And yeah, I'd love to hear
Yeah,
your
so
perspective.
I've had a couple of discussions with through the Cell and Gene Foundry that I moderate on a monthly basis. One of them was basically in cell therapy, the product is not the process. The product is the product. Especially in autologous, you start with a patient material, and by definition, that starting material is different from one patient to another.
Hmm.
Um, And at this stage of the industry, it creates a variability that you can't really fight against. Um, just for that, and that's not the only reason, but just for that reason, the process is not the product, because you can do the process 100 times on 100 different starting material, you will get a different product at the end. And the characteristics of that product will be different. What we discussed and what I've been suggesting to whoever I talk to, is really starting with the idea of product in mind. Define the product that you want, an efficacious product with what's available. Of course, if you work again, like with a CAR T, it's a little easier. You have 15 years of publications, clinical publications, on what the right phenotype for a CAR T can be, and what you should aim for when you manufacture your CAR T. For other cell therapies like iPSC based, etc., those are a bit trickier because there's not as much clinical data out there.
Hmm.
But for, let's say, T cell therapies at large, there is a lot of publications out there that inform what the nature of the product should be, from at least from a phenotypic standpoint.
Yeah.
And understanding your product, the target product profile, is critical in what we're trying to achieve in CMC, because it's only by knowing the target that you'll get there. Then you can engineer your process around it. I've seen over the last few years many companies, whether they're therapeutics or device, um, putting out there some data, and they're just publishing data that does not match what the clinical publications tell us the profile should be. I've published a few articles recently on that very concept of looking at the product and the phenotypic profile of those products and why it matters. And for example, the memory profile of the T cells seems to have a very critical impact on the outcome in patients. The more stem cell memory T cells you have, the better chance of a positive outcome you have. The more effector memory T cells you have, the less chances you have. It's not black and white.
Hmm.
It's biology. It's very likely never going to be black and white. But that's why you have statistics.
Yeah.
You play with those numbers and you draw conclusions based on that. Here, if we take the example of the phenotypic profile, I see many different companies using, in what my opinion, the wrong device, the wrong strategy. They just manufacture fast at low cost, which is great. COGS are important, but if you manufacture a COG-friendly product that is not efficacious at the end, what's the point? You're not gonna treat patients. You're not gonna have positive clinical data. the company's going to end in 2 years. Yeah, you made it to phase one, great. But then there's nothing after. There's enough data out there now that we can actually shape that idea of a product first, and then engineer the process to make that product.
Okay, that makes sense. So what seems to be sort of unique about cell therapy is this difference between patient responses. And so that makes a lot of sense that the process isn't the product because of the unique response of different patients. So I'd love to sort of clarify a bit with the product definement. We're talking about phenotypes here. So does that mean you have a defined phenotype the patient must, like, this is how it must work inside a patient, and now fine-tuning process development on a sort of per patient basis? Is that how that works?
No. There's ways to, for each unit operation of your manufacturing process, there are ways to influence how the product will evolve. So for example, let's take the memory profile again. Some patients, depending on how many lines of treatment they had before, will have different percentages of TSCM, the stem cell memory, the central memory, the effector memory, and the differentiate effector. Those are the four main categories. Depending on the lines of treatment, the starting material might be widely different from a phenotypic standpoint, but there are ways during the process, the cytokines, the media that you use, the bioreactor that you use, that you can tweak those. It's basically, so the whole logic of it is that you define your product, the quality target product profile, and then from that you derive the critical quality attributes. Those are, for example, the memory profile. And from those quality attributes, you derive critical process parameters. And those are what you're going to be able to act on to tweak, change your CQAs that will end up changing your QTPP. For a bioreactor, for example, you can tweak the dissolved oxygen. It seems like it's a parameter that has an impact on the memory phenotype. If you're 100% oxygenated all the time, you might end up with a more effector profile, less stem cell memory, for example. If you're in a DO that is around 30 to 40% through the culture, you end up seeing a lot of stem cell memory profile. So those are actionable items that you can play with to ensure that no matter the starting material, or as much as biology allows, you have ways to tweak your process, make a general process to ensure that at the end you have the quality target product profile that you have.
Okay, so it sounds like these process parameters throughout, as you are developing, these become the dials that you are allowed to adjust as you are moving through. So does that mean, as like, in order to deal with the differences between patients, when you are defining these CPPs, those are the things, for example, like you mentioned, dissolved oxygen, that is something you could change between patients to adjust for differences between them?
I wouldn't, because you have to define your process. And once you submit your process to the regulatory agency, right, it's pretty safe. I think the future will come to more flexibility to allow for that, but at this stage, at least with the projects I've worked on, process doesn't really allow that. That's why you need to really optimize your process and make it as homogeneous as you can, and work the variabilities, engineer the variabilities out of the product by defining your process. If that makes sense.
Yeah. Now, do you see these challenges as sort of unique to cell therapy, given that it's like one delivery to one patient as opposed to like generics where you're manufacturing a huge amount and it's the same for everyone?
Likely, I've never worked with small molecules or antibodies personally. So my expertise in that is very limited. But it seems to be very cell therapy, autologous at least, autologous cell therapy centric. And of course, like now we're seeing more and more allo and more in vivo ideas out there. Those should work more like something off the shelf than autologous, where you need the patient material, of course, to make your product. But again, those come with a certain level of complexity that are probably going to be very um, relevant to those modalities. Again, like we talk about cell and gene therapy at large, it's a very large bucket. Um, I'm not entirely sure you can take all the learnings from cell therapy and apply them to gene therapy.
Yeah.
Those are very different modalities. So same for the other types of products.
Yeah, that makes a lot of sense. And yeah, I've heard that it seems that there's a sort of emerging wave of these in vivo delivery vehicles. And it seems like that is one of the aims, is to make it a bit more off the shelf in that sense.
Yeah, absolutely.
Great. Well, I'd love to talk a bit about the tacit knowledge gap. So something that's come up in a number of conversations with the cell and gene therapy world is documenting processes is challenging, and there seems to be a lot of technical nuance that can be a burden during tech transfers. So I'm curious if you see this, or if you see it sort of as the technologies are standardizing, if they're becoming better documented, and so there's less of this problem.
Yeah, so it's unfortunately still a core issue in our field. Um, I think if we want to succeed as an industry, we need to minimize as much as possible, annihilate in an ideal world, any notion of secret sauce.
Yeah.
Um, Having gold hands at your
Yeah.
process and be the only person who can make it, well, you don't have a product.
Yeah.
You need to document your process so someone else, a CDMO likely,
Hmm.
can do it reproducibly every time. Otherwise, you just don't have a product. It's as basic as that. So tacit knowledge in an academic lab, fine. When you start making products, that should be something you want to avoid at all costs. This is a source of variability that should not be considered. It's something that needs to get out of people's minds, people's behaviors, people's culture.
Yeah.
Someone else in another country on the other side of the world, around the corner, should be able to make as reproducibly as possible.
And so something else that's sort of come up in this is it's not just having the golden hands, but also the troubleshooting expertise. So, if you're a PhD who's spent 5 years developing this, you know the ins and outs and how to do everything in a very perfect way. Whereas when you're handing it to a CDMO, this is no longer a research. It may not be a PhD in this area. It is now a factory. And what you are handing over this to a factory worker to run their 8 to 4 shift. And that is like not just a different mentality, but it is like a whole different mode of operation. So
Mhm.
yeah.
Yeah, absolutely. And that's why the tech transfer is so critical. And a huge part of the tech transfer is the documentation around your product.
Yeah.
Um, I'll take an example from when I was working at a CDMO. We had a client who was using a mix of IL-2 and IL-7 for their process. They were using non-GMP IL-2. We were using as a CDMO GMP IL-2.
Mm-hmm.
Nobody noticed that they did not use a GMP IL-2 in their process in their lab. So when they sent us cells and we reproduced the process with our GMP IL-2, we had wildly different growth curves, and no one could figure it out. It's like we have the same process and the same materials. Well, one day on a call, someone, instead of saying IL-2, said GMP IL-2, and the client, on the side of the client, someone was like, wait, we don't use GMP IL-2. Would that have an impact? Guess what? Of course, the GMP IL-2 has a very defined concentration. The non-GMP IL-2 that they were using did not. It had a range. And of course, the concentration of IL-2 was the key factor. They moved to GMP IL-2, they did their test, everything matched. So the way you document your process, you characterize the product at each step, you qualify your assays. Your assays at the end of the day are what matters, because that's the best way you have to follow the evolution of your process and your product. So you have to have everything well documented to start your tech transfer. The CDMOs have expertise in making your 10-page protocol, if I'm being optimistic, into a 300-page batch record. That's part of their expertise, and that's fine. The batch records takes into account the facility specifications, etc., where you have a process that you've done in your own lab for a while. But the details of that documentation, of that batch record, that's what's going to allow you to engineer out all those variabilities due to the secret sauce that should not exist.
Yeah. So something I'm curious about is so the story you mentioned with the IL-2 going from like a research grade to the GMP, realizing that was a cause of variability, then going back to the research environments and engineering it out. Do you see this sort of back and forth happening a lot during tech transfers when people are, they're going into GMP manufacturing?
It depends on the maturity of the product development team. Usually on the therapeutic developer side, you have teams with people who have been in this for 20, 30 years. Usually you don't see that. When it happens is when the companies are young, usually doing it for the first time, and they try to cut costs as much as they can to get to the next round of seed funding. And one of the easy ways is to cut on materials. But unfortunately, there's a reason why we use GMP materials, and why I would always advise that whenever you do process development work or product development work, excuse me, you use GMP materials as much as you can, because that is going to fast track your tech transfer wildly.
So thinking sort of long term about the life cycle of these biotech companies, what is the sort of taking a higher level standpoint? What is the impact on the business of this? Like these delays happening from tech transfer, figuring out and engineering out these quirks we're talking about, like the IL-2.
Yeah, for sure. I mean, CDMOs are not cheap, and there's a reason for it. It's a very well controlled environment that costs a lot of money to operate. It's high qualification employees usually that are operating the quality team, the manufacturing team, the QC team. So operators are usually fairly expensive, especially in the tech transfer and the management, because there's a lot of pressure from a regulatory standpoint on those teams.
Yeah.
The facilities are expensive to run. You have extensive air handling units that need to run 24/7, 365 days a year. And that is not cheap. So those operations cost a lot of money. So CDMOs are not, they're expensive, but they're pricey, but they're not extremely expensive. Like
Mhm.
there is a value to the price that CDMOs offer. Um, Anyone who tried to build their own GMP facility in 2020, 2021, when money was free,
Mhm.
realized very quickly that, yeah, it does cost several $100 million to build from the ground up. So when the CDMO is already made, well, it costs a lot of money, and it costs a lot of money to build, and it costs a lot of money to operate. So any delay, whether you talk in months, in weeks, in days, any delay is very costly delay.
Mhm.
And that fee will be pushed back to the therapeutic developer in one way or another.
Yeah.
So if you do the groundwork before you start your tech transfer
Mhm.
and get as tech transfer ready as you ever can with the money and the space you have and the people you have, it's the cheapest way. Because once you start your tech transfer, you can do a tech transfer within. Tech transfers usually are anywhere between 6 and 12 months.
Okay.
That's a general window. It's explained by basically you have to do all the documentation, transforming your protocols into batch records for the manufacturing, transforming your protocols into assay,
Mhm.
GMP assay protocol for QC. You have to qualify some assays, you have to validate some assays, especially the safety ones. Um, and then you have to train new people. You usually don't train on a process over just one run. Takes a couple runs to make sure that people understand that the batch record is written properly. You can write a batch record in a silo and then you try in a room and it's like, oh well, that doesn't work. The flow doesn't work. So you do modifications. You do a couple of runs for training and batch record optimization. And then you go into what people call either GMP runs or engineering runs, where
Mhm.
basically it's your IND enabling runs. You need usually the ideal number is 3 that you put in your regulatory package. Sometimes there's negotiations where you can just have 1 with the promise of doing another couple after the submission of the IND. But those take time because one run is usually about 10 days, give or take. Again, when you keep the CAR-T model, it's about 10 days. You have at minima 2 or 3 days of release. So end to end, you're about 2 weeks. And those ones, you can't really overlap them because you learn from each of them to hopefully improve the next one. So it does take a certain amount of time just for that. The documentation piece is huge. Sometimes it takes 3 months to just write those documents. Um, and then the submission of the IND. So all in all, anywhere between 6 and 12 months. The fastest tech transfer I've seen and I've operated was 4.5 months.
Wow.
Um, and that was extremely fast, extremely stressful. But thankfully, the therapeutic developer knew their product and their process. So they had done the documentation ahead of time. The tech transfer was a lot easier than in any other cases. The documentation was very GMP ready.
Okay.
Um, so it was very easy to transfer, but most people, that's just not the reality, especially if the company is young.
Okay. So that case, the 4.5 month one, that was very well documented. That's
Absolutely.
that's the way you can really accelerate this tech transfer is have it all to a T and engineered.
Yeah, no, no. You're the owner of your product.
Yeah.
You're the owner of the process. it's your job in a way to know it and be and make that package as GMP friendly, as CDMO friendly as one can. If you don't do your homework in the first place, well, the tech transfer, you can't blame the CDMO for a poor or a long tech transfer if you haven't done the work in the first place. If you've done the work in the first place and the tech transfer doesn't work, then that's another story. But if you haven't done the work ahead of time, it's just a bit complicated.
Yeah, well, this is a story that is unfortunately not the first time. This is
Of course not.
this is common. It happens.
And that's one of the reasons why I'm doing what I'm doing. Um, I do want to help these companies. I've worked at a CDMO, I've worked in at a therapeutic developers, I've seen both sides, and over the last couple of years of consulting, most of my clients are around that phase, and knowing what the expectations are down the line allows you to work ahead of time and make those changes to avoid that 2 month delay in your timeline. Because of course, like that might be a delay that is just a week, but that week very quickly becomes a month when you add, oh, this created this error created an
The
out of
dominoes.
spec result. You need to study this so we don't make the same mistake later. Well, guess what? That 2 day assay error became 2 weeks of investigation and then another week of documentation and then oh you missed the next slot, so you have to push by a month quickly or like 6 weeks 8 weeks down the line because of something that happened during one of the assays, for example.
Yeah, well, this
um,
seems to be just a general theme in any complicated process is when you have these minor mistakes, they quickly domino,
Absolutely.
just as you described. So one thing I'd love to sort of dive a bit deeper into is the engineering runs. So
Yep.
how do these engineering runs fit into this? Like, what is the technical purpose, I guess, of these engineering runs and the regulatory purpose? How do they actually fit into this?
So the engineering runs, or GMP runs, the way some people call them, are to show that you're in control of your process and your assays. Um, so you follow a finalized batch record, you use the final material, you go end to end. So if you have like a patient-based product, you're going to use a healthy donor material from the get-go, full leukapheresis, going through the motions as you will during the manufacturing campaign during your clinical trial. So you don't cut corners, you do full end-to-end, you use qualified and validated assays for the in-process and the release assay in QC. You do the full documentation review, you release your product, you go through all the motions to show the regulatory agencies that you can do what you said you would do.
Okay. And so, we're talking about the documentation and sort of how the better documented you have your process, the less time it takes. So one thing I'd love to understand more specifically is, are engineering runs one of the places that plays out? Like if you have not done your documentation as well previously, is it repeated engineering runs? Is that one of the causes of these larger tech transfer timelines?
So technically, when you kick in the engineering runs, your batch record should be as finalized as possible. Of course, as we said, like before the engineering runs, you may have had 1, 2, 3 training/pilot runs.
Mhm.
Reality is that if you don't have a process that is well defined, surprises will happen, and it can happen during the engineering run one. Um, I don't know if we said that we were going to use a healthy material from the start. Some companies use donors that they know, and that's usually a smart thing to do.
Mm.
Not know the person, but know the material. Um, but sometimes you can't, and you have to deal with an incoming material that will be, I don't know, extremely monocyte rich
Mhm.
compared to what you've been working on before. Well, surprise. That's going to have an impact on your process. Is that going to prevent you to make those? Maybe.
Mhm.
Um, that engineering run one might not work out as an engineering run. It might become a fourth pilot run. And
Yeah.
then what you had planned to be your engineering run two becomes your engineering run one. You have to do one, two, three again. So you get some delays. Unfortunately, no one necessarily in cell therapy has the luxury of doing 20 pilot runs to make sure that we train on all eventualities.
They're
But
expensive. Yeah.
they're expensive. They're very expensive.
So that like that particular story you just shared of the monocyte rich donor, like that sounds exactly like one of those controls, one of those things where if you have characterized how you deal with different, monocyte concentrations, if you're when that happens, um, you're gonna have the knowledge and the control to be able to deal with it. Whereas if you haven't done that process characterization work, you're gonna be in the dark.
Yeah, absolutely. And to be fair, like you only know it can happen when usually it happens to you.
Mhm.
Like at Tramvia before, like we had a very well characterized process. Went through, I don't know, accumulated 100 runs between product development and clinical manufacturing. Um, we had very high manufacturing success percentage. But one day, one of the patients showed up with an extremely high monocyte concentration,
Mhm.
and that derailed the run. We managed to make those, but by nowhere near the margin that we were used to. Usually, we're able to make two, three bags. We barely made one. Um, so a huge impact on the run, and the root cause that we had established at the time was that monocyte concentration can happen. That was probably patient, I don't know, 11. So we had done already like 6 or 9 months of work already in the clinical setting. Things happen. You can't plan for literally everything, but
Mhm.
it's by using those people who have accumulated experience over time that you can learn to try and plan for that and make the most robust process.
Yeah, that seems to be a general theme in engineering is you don't know what to account for until it has broken. So it makes a lot of sense that that expertise is, yeah, it's what accelerates that. Well, yeah, it's been a very insightful conversation, and it's certainly shifting my thinking more towards product orientation rather than process orientation.
Hmm.
And it really shows the sort of specific value of that for cell therapy, given this high variability. Now, as we're sort of wrapping up, I'd love to know if you have any sort of key takeaways or advice for these founders who might not have done this before, who might be approaching their first tech transfer, they're in the R&D, pre-IND phase as we're wrapping up.
Nice. Let me get my brain in order for that one because that's a big one. Again, bears repeating, focus on the product, um, because that's what you're going to put in your patients. Try to know as much about your product as you can. Um, Again, I get it. Like people don't have the money that they used to have. A lot of the work has to be done on the cheap side, but it's fine. But try to spend your money smartly. Um, be GMP ready so you don't bet on a successful tech transfer when you didn't put your chip in the right place. Be product focused. Don't underestimate the value of analytics. The analytical assays are the only way you'll have to define your product and to know where you're at with your product, with your process. If you have a poor assay, you'll have a poor product definition, and it's not going to help. We talked about tech transfer from a manufacturing standpoint a lot. From an analytical standpoint, you're going to have to transfer those assays. You're going to have to qualify those assays. These take time. Honestly, more often than not, longer than the actual process tech transfer.
The
Because
analytical more
in
than the
many,
process.
yeah, because in many cases, those assays will have to be developed and qualified. Qualification takes time. And in some cases, especially in later stages, phase 2, phase 3, you're gonna have to validate those assays.
Mhm.
And validation is a beast of its own. Speaking about thinking ahead of what can happen, that's the whole purpose of the validation on the assay side is you're going to have to know exactly what can happen and make sure that you engineer out those potential variations. So your assay always works the way the assay is supposed to work. The results is a different issue. That's why you develop, qualify, and validate those assays. So don't underestimate the timeline on the analytical assay and whatever you put in there. Try to be as GMP as you can before you take transfer. That saves surprises. And honestly, like the tagline at the end is like, you're the owner of your product. At the end of the day, you can blame CDMOs or other services as much as you can, as you want. You're the owner of the product. It is your role to make sure that the product is transferred, manufactured the proper way. And if you take that ownership at heart, you'll do the work because you don't want to waste your own time and your own money. Um, and talk to your network. There's dozens of people out there who have done it, who ended up tripping on that little trick.
Yeah.
And once you're hurt, once you don't forget.
Yeah.
So that goes in the long-term memory, and you keep reusing it over time. Those are get advice early on. It's fine not to know everything. Just go get the expertise out there. It exists. People have
Mhm.
done it hundreds of times.
Yeah, I mean, repeating mistakes is yeah, it seems to just be a waste overall, and doesn't make a
Yeah,
lot of sense.
and on the scale of the industry, it's crazy. There was another webinar that I attended a few weeks ago where one of the biggest cost from an industry-wide perspective is that companies keep making the same mistakes over and over and over. They don't talk about it.
Hmm.
They don't teach the rest of the industry that they made those mistakes. So everyone keeps on making them again and again and again. And from an industry-wide perspective, it's millions and millions of dollars that go to waste because negative results or general knowledge, call it the way you want, has not been shared properly. Um, it's fine. People make mistakes. It's research at the end of the
Yeah.
day. Like clinical research is still research. Um, there are mistakes and there are errors that are going to be made on the way from cost standpoint, for example. I use this product. This product's terrible, but I'm not going to tell anybody because I made the choice in the first place. Well,
Hmm.
tell the world. It's fine. You tried it, didn't work. You know why it didn't work, and that's a faulty product. Tell the world. So 10 other companies don't use that product. It's not great for the person who manufactures that product, but it's better for the industry at large.
Yeah. So one thing I see this a lot. There's a general sort of cultural trend here where there's sort of the siloed operation, and especially through talking to CDMOs, I hear consistently that everyone thinks their product and process is extremely unique, they're often not that, from the CDMO standpoint, they're often not that unique because they actually see tons of them. And I also think this is just a waste that you have so many people spending so much time, energy, and resources making the same mistakes. Now, one kind of question I have is this an IP problem? Like, because of,
Oh,
is that why this exists? And where's the barrier to that? Is it, like, for example, product choices? You mentioned, like, that example you mentioned, I've used this product, it didn't work. Yeah. How do you see that?
yeah, no, that's a good question. To be honest, I'm not the best expert to talk about this. That being said, I would redirect your listeners to the November episode of the Advanced Therapies Think Tank
Mhm.
webinar, where that was basically the core question.
Okay.
So go check it out. It's about an hour long. It was a very lively discussion and very interesting. So if you're interested in that topic about IP fragmentation and how it kind of like hinders the industry as a whole, go check it out. It's a really good episode.
All right. Well, thank you for sharing your knowledge on the episode today. Yeah. Thanks a lot.
No, of course, it was fun. Thanks so much.